Then to think about the gene duplication, and I want to illustrate, just highlight the questions by Zhang in 2003. Because after all the study of gene duplication, I already told you it seems there are advantages to have duplicate copy in the genome. And you will also see people look at the plants and look at tetraploid, compare tetraploid and the original diploid ancestral species and compare the fitness differences. And also in early days, people even study or compare the allopolyploidy and the autopolyploidy, and so and sort of propose the allopolyploidy contain more genetic diversity and have better fitness compared to all the ancestral species. So you can see all these kind of studies seems point out the advantage of having a duplicate copy. Then in some of the cases, we also say, in the duplicate lineage, we observe accelerate evolution. So it means the positive selection acting on that specific lineage. But maybe we should ask how often this happened, and also what is the relevant, relative importance of this type of a Darwinism selection acting on duplicate copies, either of the duplicate copies. And compared to what? Compared to, for example, purifying selection, genetic drift, all these evolutionary forces co-occurred in the population, and why the Darwinism selection is more obvious to most of us. So we can think about that question, and also how this is important for its functional divergence. So I want to remind myself here and remind everyone as well, in the Force et al. Model, the gene duplication, the phase of the gene duplication, they focus on the change of the cis-regulatory region, because it's sort of a new idea and relatively. But one thing we have to remember or keep in mind is, once the function diverge between the duplicate copies, the coding sequence will diverge as well. So in that slide, the coding sequence looks the same, but you can think about, given enough time, the coding sequence will diverge as well. So this is the first question, maybe important in this field. The second part is how the new function originated and how it means, because in the Force model, we see the cis-regulatory change could lead to functional divergence, either in neo-functionalization or subfunctionalization. In the retrotransposition part, it's also important to acquire part of a cis-regulatory element to become a functional copy. So this is the part I think we all can think about it. And sometimes it's not that easy to figure out all the process, because you have to recognize the newly originated genes, like studies by Manuel Long's group. They discover new genes in Drosophila lineage, and using a comparative study between close related species, and try to figure out how the new function was acquired, was evolved in a specific lineage. The third one is, if we look at a gene or a gene family which composed of complex expression pattern, and how this can be resolved. Why we say that? Because think about a gene have multiple cis-regulatory modules. And we say, simply in the model, we simply say, oh, each cis-regulatory module enhancers, then they specify the expression in certain tissue or in certain development stages. But think about in development, the well-studied cases. For each enhancer, there are several proteins, several transcription factors, specific transcription factor bind to this region. And this transcription factor also was regulated by other transcription factors. So in the case, in the organism, in a cell, even in a single cell, the regulation is very complex. So nowadays we like to talk about gene network. So one of the layer will control the bottom layer, but there's also a lot of interactions among them. So the network itself, after the duplication, how this new copy fit into this new network. It has to have transcription factor bind to, it has to act to downstream target. So this is not a very simple case saying a gene acquire promoter, so they express. There are also other things they have to cooperate with in the single cell. So the protein-protein interaction is really an important part we have to think about, because no gene acts alone in the cell. So it has other genetic partners in the cell. So this will be an important process. Because now we study genes, we only study the evolution of those genes, but sometimes we have to think about the evolution of the entire pathway. And the other is the buffering. We haven't really talked about. We just say, okay, a gene duplicated by unequal crossing over or by retrotransposition. But think about if the dosage is important. Next week we're going to talk about sex chromosome evolution. We'll talk about a lot of things on dosage. Because in males, in human, males have only one X chromosome, female has two X chromosomes, and we have specific mechanisms to act for dosage compensation. And think about in a genome, there's extra genetic material originated by a mutation. Then suddenly the individual itself originated genome has one copy of this gene. Now we have two copies. So the cell, first thing the cell has to do is it has to be able to tolerate the dosage differences. If both genes are expressed right after the mutation. So the model itself, or the model we talk about, is very simple. We haven't really touched on how this dosage was tolerant, or what we're showing here, what's the buffering system, how our genome can tolerate the duplicate copy, and how the genetic buffering evolve, or how this be important part of the duplication process. And the last part is the speciation, is how the gene duplication related or be important to speciation. Actually, I don't have extra time. I was planning to talk about some of my own observation and research on the gene duplication and speciation, but a lot of study now showing at this certain proportion of the genes related to species differentiation are the new gene materials. So new gene material means the gene is a duplicate copy, or the gene contains duplicate parts of the exons. So this is really important. And how important is this observation? We don't know yet, because we have limited knowledge on how many genes involved in the speciation process. So this will remain for future study to allow us to further understand the importance of gene duplication and in speciation.